Okay, so now we move from the reassuring information, our data that Giuseppe provided, to a worst case scenario, meaning that SARS-CoV-2 can readily replicate in the brain of susceptible mice. And I will tell you a little bit about why this worst case scenario is actually important for us to find out more about the virus and its pathogenesis. So Long COVID, the project, combines clinical and mechanistic studies. And we today talk about the mechanistic studies other than those on clinical samples. You've already heard Giuseppe, who talked about the in vitro studies on human neurons. And this is our part here in Zurich. It's about the pathomechanisms of neural infection and in K18 ACE2 mice. I'll tell you about those. Meaning we use experimentally infected mice to really examine the direct effect. What are the benefits of such mechanistic studies? We have very defined experimental conditions and can thereby eliminate any confounding conditions. Giuseppe mentioned comorbidities. We could think of co-infections. And of course, in any patients, we will have no idea when they became infected, what the dose of infection was, etc. So it's the stringency of the experiments that allows detection of slight but potentially relevant effects, because we use basically a set of almost identical animals, a defined dose of infection, a defined way of infection or challenge, the intranasal one, and we know precisely the time point after infection that we examine the animals. So I don't need to tell you much more about COVID-19 and the brain infection in the patients. So there has never been any SARS-CoV-2 viral particle identified at ultrastructural level. That's for sure, but as Giuseppe said, there were traces found in some fatal cases. Now, what would normally be the route of infection of the brain that you would expect in patients, considering you're looking at a respiratory virus that heavily replicates in the nasal cavity, is the olfactory route. However, this is really not common in COVID-19 patients. The olfactory epithelium, which is the one responsible for, starting off, I should say, the smelling, becomes infected. There is loss of cilia, there is a disclamation, but interestingly enough, then only that's a difficult word, sustenticular cells, support cells of the epithelium become infected, but not the olfactory sensory neurons. And these neurons would be the cells that get the virus into the brain via the olfactory bulb. So apparently, these cells are at least generally not infected. And the loss of smell is due to infection of these sustenticular cells. However, we ask the question, what could happen if limited brain infection occurs? And for that, we needed to use an animal model. So wild-type mice are basically resistant to infection by most SARS-CoV-2 strains. They express murine ACE2, the receptor that Giuseppe already mentioned, and the viral spike protein binds very poorly to it. However, the susceptible mice that I mentioned before are transgenic mice that carry the human ACE2 receptor. As soon as keratin 18 is produced, human ACE2 is produced as well, which means that this receptor is expressed in epithelia, including airway epithelial cells. And therefore, intranasal challenge of these mice with the virus allows rapid infection. And different from humans, infection of the olfactory epithelium leads to very frequent spread to the brain via the olfactory bulb. And you can see this here. Whenever I show you the pictures with the brown here, that means we show the viral antigen. And also a difference is that compared to humans, mice have a huge surface area of olfactory epithelium because their sense of smell is vital for them, of course. So let's have a look again. SARS-CoV-2 infection in the ACE2 mice is accompanied by a mild inflammatory response. You saw the brain before. You can see there's a really widespread infection. And it's just the cerebellar cortex that's almost spared infection here. What we can see is that there is a limited inflammatory response that I mentioned, represented by perivascular infiltrates of leukocytes. And you see these, as well as the vessels, are completely spared of viral antigens, so these are not infected. How does it look like? For this, we used transmission electron microscopy, where we can go in very high magnification in very thin sections. I explain that in a few minutes. So imagine this infiltrate here. If we go to the high magnification, you can see a vessel. You've got the red blood cells, and you've got the lining cells, which are labeled as E, the endothelial cells. In the lumen, you have monocytes and neutrophils. These are the main leukocytes that play a role here, plus some lymphocytes. And they sometimes attach to the endothelium, or like here, have already left the vessel and are in the perivascular space. A closer look shows this here. For example, you've got a neutrophil that's just leaving the vessel and it's squeezing itself through a very small hole in the basal lamina. And here we've got a monocyte that has left the vessel already. It's underneath the endothelial cell layer, and this is the perivascular space because it aligns here to the processes of the astrocyte, so-called astrocytic end feed. Now, these leukocytes, they migrate to some extent in the tissue, and you can here see that in T lymphocytes and in neutrophils. And there is, this should ring a bell to what Giuseppe talked about. We have the microglial cells. They become activated. You see them as plump amyloid cells here with a marker for the microglial cells, and they replicate to some extent, forming small microglial nodules. So we call this microgliosis. Okay, we see a mild inflammatory response, but it does not target infected neurons. Here you see all these neurons expressing viral antigen massively, but the leukocytes are not infected and are not aggregating around them. There are no dying neurons, at least from what we can see here. And I show you again the picture of the T cells and the neutrophils. They seem to have gone into the parenchyma, but are a bit lost in space. They are not targeting anything. So we then used bulk RNA sequencing to actually gain some information that goes beyond the description of the inflammatory processes. So what you can see here, we've got samples taken from the thalamus, which is the area where we see the most intense, if at all, inflammatory response. The volcanoplot on the left shows you all the genes that are upregulated, meaning being transcribed and sort of wanting to become proteins that are then effective in the cell. In total, there is more than 1,000 genes upregulated in the infected brains where we find the virus. We then had a look, a more in-depth look into using a gene set enrichment analysis, meaning we wanted to know about the inflammatory response. And here I can show you the top 20 upregulated inflammatory mediators. And there are a range of cytokines in there, plus some evidence of innate immune response. I come back to that later. We then used pathway enrichment analysis because that allows us to really look at specific pathways of interest. And here we've got the inflammatory response using the CAC analysis. And you can see all the red ones, they are high, they are upregulated. And these are a lot of chemokines and cytokines, here the TNF-alpha family, all related to the inflammatory response. And this one I find particularly interesting. This reflects leukocyte recruitment because here we looked at pathways that are relevant for leukocyte transendothelial migration to leave the vessel, like what I showed you here for the neutrophils. And again, you see a range of relevant genes upregulated here and pathways, of course. So what can this worst case scenario that I showed you tell us? First of all, when you see this, you wonder, you see a relatively mild inflammatory response. You see no dying neurons, at least not at first sight. We have another close look into that. So the hypothesis is that probably SARS-CoV-2 infects neurons and even replicates in them without any neuropathic effect. So we then wanted to investigate the SARS-CoV-2 infection in these neurons in more depth, meaning in-depth morphological studies, and actually find out about viral replication and see potential functional changes. So for that, we used what I mentioned before, transmission electron microscopy. And here you can see our positive control. These are human embryonic kidney cells that are stably transfected to express ACE2, the human ACE2. So they are perfect to reproduce a productive infection. And here you can see the viral particles in membrane-bound vesicles. So transmission electron microscopy means we take cells or tissue that we fix very well and then embed in epoxy resin, so like a plastic embedding, which allows us to prepare very thin sections of about 100 nanometers. We then contrast them and put them into the transmission electron microscope and send an electron beam through, and we can then identify structures, and that is based on the difference in their electron density. You see here the black ones, they're very electron dense, and here the light ones, they're electron lucent. So this as a control for what we look at later. So I already said there is no evidence of pathological changes in infected neurons. This is just to confirm it. So here's the viral antigen, but on the consecutive section, that's our usual hematoxylin eosin stain section, we see nothing. These neurons look perfectly happy. Yeah, so we have the impression here, because we have both spike protein and nucleocapsid protein, which are the main ones to detect, that these neurons actually carry the infection without being damaged at all. Now let's go to ultrastructure again. On the left, we have a mock-infected control mouse brain, and I try to outline here with the blue a neuron. So the unaltered neuron has a nucleus in which it carries the DNA that is transcribed to the mRNA to produce proteins later. We have mitochondria here. You can see them as these structures, or here. These are responsible for energy production in the cell. And we've got the rough endoplasmic reticulum, where all the protein transcription and then translation occurs. Now, if we look at a neuron in an infected brain, we see mitochondria that look fine. You see them here. We see RER. We see a nucleus that looks pretty happy, but we also see these structures. And for us, these are evidence of viral structures that we then investigated in more depth. Look at an infected neuron here. I can tell you it's infected. When we go to the high magnification, we find the membrane-bound vesicles containing the viral particles, as I've shown them already in the HEC ACE2 cells. And then we go to the schematic that's similar to what Giuseppe has shown you already. And you can see the HEC ACE2 cells and here a neuron in an infected mouse brain. So it's the RER where the proteins are then translated. So the nucleocapsid and the other proteins accumulate somewhere in a structure that's between the endoplasmatic reticulum and the Golgi, where we can see it as these aggregates here. And then later the virion is assembled and then delineated by a cell membrane and then released. And these membrane-bound vesicles is what we see here. And so that everyone can be convinced that these are viral particles, I've chosen to show you some immune electron microscopy pictures. So we use now the antibody that we use to detect viral antigen in the brain, and you saw as the brown stain, and used secondary antibodies that are coated with gold particles. These are these small, pretty homogeneously sized black dots, and they clearly accumulate in these areas where we think we are looking at the viral proteins and then the viral particles themselves. So we went back to the data that we had generated through the bulk RNA sequencing and really wanted to know whether something happens to the brains. So first, please have a look at the principal component analysis. That one allows you to identify the major sources or reasons for variation. And the PC1 is the one with the strongest. And anyone can see here that the infected brains, they're clearly clustered together, and so do the uninfected brains. Now, we used a sophisticated way for cell deconvolution. With the help of a reference profile for the mouse thalamus, remember our sample is from the thalamus, that comes from the literature, and using a sophisticated analytical tool, a software called CyberSort X, it's possible to actually allocate, based on the transcription pattern, cells. So please have a look here at the infected brains versus the mock-infected ones. I highlight this. It looks like the light green, the proportion is less, and so is the proportion of the dark blue, meaning excitatory and inhibitory neurons, whereas the proportion of red, meaning microglial cells, is increased. So there is evidence of some, albeit minor, loss of neurons and an increase in microglias, which is, of course, consistent with what we showed before, the microgliosis. However, there is a bit more going on, of course, and that is a strong antiviral response. We see an innate immune response. We have effectors interleukin-1, we have the type 1 interferons, but what is particularly interesting is the reaction to the virus. And when I draw your attention to this, it shows negative regulation of viral process, defense response to virus, negative regulation of viral life cycle. So the brain, or let's be the neurons, really try to do something against the viral replication. It doesn't end here. When you have a close look, you can see that there is a negative enrichment, which means, in simple words, sort of a downregulation of pathways associated with axon and neuron projections. One that isn't downregulated is the positive regulation of neuron apoptotic process, which could indicate or explain why we get the impression that a small proportion of neurons might be left, so death by apoptosis. But otherwise, it looks like the cells cannot reorganize themselves or not develop. There is like morphogenesis that's downregulated. And then we have some almost negative enrichment of pathways associated with synapses. What's interesting as well is that there is a consistent negative enrichment of some pathways associated with certain behaviors. And that would be learning, associative learning, and exploration behavior. As examples, why do I show these? Because we had a little go at examining the mice for some behavioral changes. So we have a set of mice that was confirmed at day 7 post-infection to have brain infection. At day 5, we realized that the number of inspects the animals sort of directed towards new objects in their cage was reduced. They also seem to have a shorter time looking at each of these new objects. That the difference was not significant, but overall, time and number of inspects of each object was reduced, which would indicate some change in behavior in the mice. So our model shows that the infected neurons, not directly dying, no, but they really raise an antiviral response, innate immune response, and by releasing probably also cytokines, some chemokines, we have an activation of the glial cells. And the microglial cells in particular can release quite a substantial amount of cytokines and chemokines to recruit the leukocytes. And there is probably some functional changes affecting behavior. One thing I would like to also mention is that we can use this worst case scenario model to undertake further investigations. And we really focus there on investigations on the antiviral therapeutics and some antiviral drugs, but today I want to talk about neutralizing monoclonal antibodies. They are used for prophylactic and treatment, and we wanted to know what is the use of them and what's the pharmacokinetic of those when they're systemically applied. And we have used known monoclonal antibodies against SARS-CoV-2 spike protein. And that's Ronapreve, a combination of two monoclonals, neutralizing monoclonal antibodies, human, that have been approved for COVID pre and post-exposure prophylaxis and treatment. And in a previous study, very early in the project, we could show that they are effective in experimental infections in our mouse model. So we treated them in a post-challenge experiment, 24 hours after infection. It avoids clinical disease shown in the mice by weight loss. And when you look at this here, they didn't lose any weight. Looking at the viral burden by our QRT-PCR in the nasal turbinates and the lung, at day 4, it was a significantly lower amount of virus in the nasal mucosa, and on day 6 in the lungs. The same is true for day 9 in the lungs, and then on day 9, there was obviously no virus in the brain. So they seem to block brain infection. And that is the case here. We don't find any viral antigen, no inflammation, no change. So the question was, does the treatment itself have any effect? So we just had another experiment and looked at uninfected mice that were treated with Ronapreve, the two antibodies. It was an intraperitoneal application. We did not see any substantial weight loss. Look at the green curve here. Very different from the untreated infected animals. And there were no histological changes in the organs. We then looked at the transcriptome in the thalamus. And you remember the volcano plot in the infected, untreated animals with more than 1,000 genes upregulated. Here we look at 23. We can list them. And for most of them, the function in the mice is not yet defined. There is no evidence of anything that's functional, degenerative, or inflammatory change. And the question is it because these monoclonals are in the brain? Or how does it happen? So metabolomic and lipidomic analysis point towards the same direction, that there is no effect. Let's look at the biodistribution of Ronapreve in these mice. So here you can see it for each of the antibodies, and you can always see it in infected and in uninfected treated animals. The serum, and then we looked at crude tissue homogenase, just squeeze the tissues and then look at the content by ELISA for each of the antibodies. In any tissue, there was a large amount of monoclonals, and it's this EC90 which says that it's 90% inhibition when you look at a concentration curve that you reach. When you reach this, then it's effective. So it must be effective in all the organs, at least. That's what the homogenates suggest. There is a low amount in the brain and in the nasal turbinates, lower than elsewhere, where in the tissues it can reach up to 10%. Here it's around 1%. But look at the pictures on the right. We had an immunohistology done against these two human monoclonals, and we cannot see any evidence of these monoclonals outside the tissue, outside the vessel. So there's no evidence that the monoclonals leave the vessels and go into the brain parenchyma. When we look at the infected animals, the same is true for the brain. There is something here in the vessel, nothing in the parenchyma. In the lung, we see a little bit in macrophages and in type 2 pneumocytes. So very limited tissue penetration. And that is probably to neutralize the virus in the alveoli, but no evidence that the monoclonals gain access to the brain parenchyma. So how does it now function? How do the monoclonals block brain infection? So this is the way it goes. We know from the olfactory epithelium, olfactory bulb, and then distribution in the brain. So the idea is that on day one, when we applied the monoclonals, what we know is that the intraperitoneal application leads to uptake in the serum. We could show that. It's also in the literature. The literature has also shown that the monoclonals are released into the nasal lining fluid at a low proportion, but sufficient to neutralize the virus. And we have evidence of that from our pharmacokinetic study. So on day one, where we have, as you can see here, in an untreated nasal mucosa, tons of virus in the lumen, in all the epithelial cells. When the monoclonals are released into the nasal lining fluid, they probably reduce the viral load efficiently. So there is less viral replication olfactory epithelium and probably just less opportunity for the neurons to become infected. So what's the wider impact of the biodistribution study? I mean, one is that it's important to understand the distribution of monoclonal antibodies at cellular level. So when you speak about true tissue homogenase and the tissue penetration that you can identify from that, you're not talking tissue penetration. You may be, like here, just looking at the content of your compound in vessels. And we can use the results of this study to inform the development of monoclonals for other viruses, pulmonary ones, because we could see, I mean, respiratory ones, we could see that the viral load in the lung went down, but also all the other viruses that get into the brain via the olfactory route. And Giuseppe mentioned a few. Influenza A, herpes virus, polio, paramyxoviruses, Japanese encephalitis virus, rabies virus. There's a long list. Okay, good. So I would like to acknowledge that, who's surprised, I didn't do this work on my own. The main person to mention here is Simon Deneck. He is a fully qualified veterinary pathologist who embarked on the project as a PhD student and now has the unique skill set of also being able to deal with omics data. Then we have experts in Zurich for ultrastructure pathology and for neuropathology. And we have a biomathematics expert who helps us as well. Our histology lab team is very important, and we've made use of the Functional Genomics Center. Then I should mention the collaborators, or I need to mention the collaborators at the University of Liverpool, because the animal experiments were not done in Zurich. They were actually done in the labs of Professor James Stewart in their BSL-3 facilities, and they have vast experience in this type of infections. And I have to mention Professor Andrew Owen and his team, with whom we undertake all the pharmacokinetic studies. And then, of course, let's not forget Giuseppe Balistreri and his team, where we line up with the in vitro studies and get all our positive controls and vice versa and work with their infection experiments. Finally, I need to mention the State Secretariat for Education, Research and Innovation, SERI, in Switzerland, that paid for my part of the study, because at the time we got the grant, we were not part of Horizon. Thank you.